of Semiquinone Radicals, Phytohormones and Sugars, and Pistacia vera, LCV, Kirmizi Development.
In this presentation, our goal is to offer you a quick summary of the research and conclusions. Please follow the links to view the research in further details. Thanks to all the authors and publishers for making this research public, thereby advancing science and innovation. Please like, share, and subscribe.
The main aim of this study was to investigate metabolic responses of fruits, leaves, and shoots of pistachio trees during their development.
Electron pragmatic resonance spectroscopy revealed significant increase in generation of semiquinone radicals in fruits and leaves of pistachio, while the flower cluster thinning application was conducted in relation to the control, especially at the second term of the plant material collection.
Moreover, flower abscission caused an increase in the level of phytohormones such as indole-3 acetic acid and abscistic acid in fruits at the first term of fruit samplings.
In turn, high-performance liquid chromatography analysis revealed differences in both the profile as well as the content of the soluble sugar detected in the pistachio organs.
The highest total sugar content was found in fruits of pistachio where the flower cluster thinning application was made early.
Total sugar levels were higher also in leaves and shoots of the above-mentioned pistachio plants in relation to the control until the third sampling time.
The importance of high levels of fructose in the fruits following the flower abscission was observed.
Additionally, analyses of mineral elements in organs showed that copper and phosphorus content in fruit were higher after the flower abscission in relation to the control.
To conclude, the findings of this research signal on contribution of semiquinone radicals, paramagnetic manganese ions, phytohormones, nutrients, and sugars in pistachio organs development on the background of the flower cluster thinning, which was applied before fruitification.